Now we're going to move a little bit into structure. The nice thing about the exam, if you were taking this exam 20, 30 years ago, you would have to study the structure in great detail. The exam nowadays doesn't really expect you to know the exquisite detail on structures. That goes for nucleotides, that goes for amino acids, that goes for fats, proteins, etc., etc. You do need to know a little bit about the structure of the bases so that you can differentiate them from one another if given a question that shows you some structure. So don't spend your time trying to memorize the way that the bases are structured. Try to learn the differences between them. That's where your time is going to be better spent. So there's two types of nitrogen bases that are found in nucleotides. There are purine bases and there are pyrimidine bases. There's two types of purine bases, and I want you to use the following mnemonic to remember these, okay? You want to remember pure as gold. The score that you get on this exam is going to be pure as gold. The awesome clinical career that you have after you're done taking this test is going to be pure as gold. So the purines, the purine bases are adenine and guanine, so pure as gold. So the pyrimidines then, we use the mnemonic CUT, C-U-T. C-U-T for cytosine, uracil, and thymine. Those are the three pyrimidine bases. How are you going to differentiate the purines from the pyrimidines? I like to think of it this way. Purines, if you look at their structure, you see that they have two rings that encompass their structure, each of their structures. So they have sort of a bigger structure, but a shorter name. Okay? The pyrimidines, they have a bigger name, but a smaller structure. There's only one ring in their structure. So if you simply want to remember the purines have a short name but a bigger structure, the pyrimidines have a bigger name and a shorter structure. So that'll differentiate those two from each other. And you know that when it comes to the rules of base pairing that you'll see later on, that adenine is always going to pair with thymine in DNA, and guanine is always going to pair with cytosine. So you're always going to see a purine and a pyrimidine base pairing with each other. Now, remember, in DNA, you're going to see A's, G's, C's, and T's. Whereas in RNA, you're going to see A's, G's, C's, and U's. Okay, we'll use those single letter abbreviations to save oxygen. We have global warming going on and so forth. So, adenine, guanine, cytosine, and thymine are found in DNA, whereas adenine, guanine, cytosine, and uracil are found in RNA. In order to differentiate between adenine and guanine, you should remember that there's simply a deamination step. Okay, there's a deamination step that can remove the amino group from adenine, and that can give you guanine. Now, it doesn't exactly happen that simply inside the cell, but for your studying purposes, if you could remember that the difference between adenine and guanine is simply a deamination step, that's all you need to know. So, how are you going to remember that adenine has the amino group sticking off of it? Well, because the amino group is found in adenine. They both start with A, so that'll help you remember that. And when you remove that amino group and you end up with a carbonyl group up on top of that structure, well, now you're going towards something that looks more like guanine. Something that looks more like guanine. Of course, there's an amino group poking off down here, but we're going to focus on this top group here. If it has an amino group, it's adenine. If that amino group has been somehow removed, then now we're looking at guanine. And actually, inside the cell, there's an important enzyme that aids in the deamination of adenine. If you were an enzyme that was going to deaminate adenine, what would you name yourself? Well, maybe adenosine deaminase, okay? And actually, that's the way it goes. There's an enzyme called adenosine deaminase that removes that amino group from adenine and indirectly can convert it into guanine. And that's an important enzyme because when deficient, it's going to lead to severe combined immunodeficiency. Okay, the autosomal recessive form of severe combined immunodeficiency disease. complete defect in B and T cell activity. The pyrimidine bases. Let's focus on these for a second. The pyrimidines, once again, the parent pyrimidine here, cytosine, is going to have an amino group poking up off the top of it. There's a deamination step which can convert cytosine into uracil. Notice the parallels here, the deamination of adenine to make the other purine guanine, and the deamination of cytosine now to make the other pyrimidine, which is uracil. Does this happen in the cell? Well, not typically. Unfortunately, sometimes spontaneously, you can get cytosine deamination. And that's actually a mutation that has to be corrected prior to DNA replication. Notice something down here, and that's that cytosine is found in both DNA and RNA. If cytosine becomes deaminated, it becomes uracil. Should you find uracil in DNA? No, you shouldn't. So uracil has to be corrected before the DNA can be replicated. And there's a cytosine deamination machinery that we need to speak about a little later in this review. All right, so now we've differentiated cytosine from uracil. Let's look at the third pyrimidine here, which is thymine. What's the difference between uracil and thymine? It's simply a methyl group. Now, this is an important methylation step. The methylation step that occurs here is going to be catalyzed by which enzyme? It's thymidylate synthase. So thymidylate synthase is going to take DUMP and convert it to DTMP. So the net effect is that you're converting a uracil into a thymine. Now, it's within the nucleoside structure, the nucleotide structure, but for our purposes here, we could differentiate uracil from thymine simply by that methyl group. Okay, so this methyl group poking up off the end of the molecule signifies that this is a thymine residue. This was catalyzed by thymidylate synthase. Do you remember what inhibitor we just talked about that could inhibit thymidylate synthase? It's 5-fluorouracil, 5-FU. So very good. Now, what is the difference between a nucleoside and a nucleotide? A nucleoside, then, is when you take one of these nitrogen bases and you bind it to a sugar. So that's a nucleoside. A nucleotide is when you take one of these nitrogen bases, you bind it to a sugar, and then you add one or more phosphate groups. So understand the key difference here. A nucleoside is a nitrogen base plus a sugar, whereas a nucleotide is a nitrogen base, a sugar, and one or more phosphates that's been attached to that particular structure. So shown here is the structure of the nucleosides, two nucleosides in particular. Remember, the nucleoside is when you take one of the nitrogen bases we just discussed, one of the 5 nitrogen bases, and you attach it to a sugar. Now, there's 2 different types of sugar you can attach it to, which we'll go into, but a sugar plus a nitrogen base equals a nucleoside. So shown here is the structure of a couple of different nucleosides. Remember that a nucleoside is when you attach one of the 3 nitrogen bases we talked about to one of the sugars. There's actually 2 different sugars that you could attach it to, and that dictates whether you're talking about DNA or RNA. So shown here is a nucleoside. In particular, this is adenosine. Now, how did we know this was adenosine? Well, looking at the nitrogen bases, what do we see? We see a big structure. So it's a short name, so you know that it's a purine, right? The big structure is the short name, and what's poking up off of this? It's the amino group. So remember that the amino group in a purine is going to signify adenine. You don't see that carbonyl group, you just see the amino group. So this is adenine. When you attach adenine to a ribose sugar down here, you don't call it adenine anymore. Now we call it adenosine. So the base is adenine. When you attach it to the sugar, we now call it adenosine. Over here on the right, we have deoxythymidine. We'll talk about the deoxy part in just a moment, but how did we know this was thymidine? Well, it has a short structure, so it has a big name, making it one of the pyrimidines. What are the 3 pyrimidines? Remember your mnemonic CUT, C-U-T. How do we know whether it's C, whether it's U, whether it's T? Well, we see this methyl group right here. Because it has the methyl group, we know that it has to be thymidine. Remember that thymidylate synthase step that methylated uracil to form thymidine? So that's how we knew it was thymidine. How did we know this was deoxythymidine? You have to look at one of these specific carbons. There's not too many carbons in these sugars that you have to focus on, but this is one that you have to remember for the exam. And that's the 2' carbon. The 2' carbon, if it does not have a hydroxyl or an oxygen attached there, we call it deoxyribose. So when you attach a deoxyribose sugar to one of the nitrogen bases, we call it, in this case, deoxythymidine. Because you attach the deoxyribose to thymine, we now call it deoxythymidine. Notice that it's always the 1' carbon of the sugar, whether it's deoxyribose or whether it's ribose sugar, it's always the 1' carbon that's going to attach to the nitrogen base. Looking at this further, you notice what we have on this particular figure is a nucleotide. How did you know it was a nucleotide? Because it has a nitrogen base, it has a sugar, and it actually has a phosphate group attached. It has one phosphate group attached, so we call this uridine monophosphate. How did we know it was uridine monophosphate? Once again, always have to go to the nitrogen base. What is this, a short or a long structure? Well, there's one ring, so it's a short structure. That makes it a pyrimidine. Short name, big structure. How did we know this was uracil? Do we see an N poking off? No, we don't. Do we see a methyl group poking off? No, we don't. So it has to be uracil. So uracil attached to a ribose sugar is called uridine. And attached to one phosphate, we call this uridine monophosphate. Let's look at another one. Over here, we have a big structure, so it must be a purine. So the question is it an adenine or is it a guanine? Well, we see an amino group poking off down here, but we're interested in the structure up top. Is there an amino group poking off up here? No, there is not. So it's not adenine, it's therefore guanine. So this nitrogen base is guanine. When it's attached to the 1' carbon of a deoxyribose sugar, we call it deoxyguanosine monophosphate. So always focus your attention on two places when you look at one of these structures in case you're asked a question with one of these in the question stem. Look at the 1' carbon of the sugar and examine which nitrogen base is attached. Figure that out. And then your second step is to see which moiety is attached to the 2' carbon. Is it simply a hydrogen, which is unmarked, or is it a hydroxyl group? If it's a hydroxyl group attached, well, then it's ribose. If there's nothing attached, i.e. a hydrogen attached, then it's going to be a deoxyribose group. So shown here is the biochemist's favorite nucleotide. You heard it millions of times. It's ATP. So ATP stands for adenosine triphosphate. Where does it get its name? Well, let's take a peek. All right, so adenosine triphosphate. The first thing we're going to do is see what's attached to the 1' carbon of this sugar. Well, is it a big or a little structure? It's a big structure, so it's a short name, so it's a purine. Is there an amino group poking off? Yep, you betcha. There's an NH2 poking off here, so there's an amino group poking off. That makes it adenine. Now, when you attach adenine to a sugar, we call it adenosine, but is it adenosine or is it deoxyadenosine? Well, that's when you look at the 2' carbon and you notice that there's a hydroxyl group there. Because there's a hydroxyl group there, that makes this the sugar ribose. So attaching adenine to a ribose makes adenosine. But furthermore, attached to this ribose is not 1, not 2, but 3 phosphate groups. So this makes adenosine triphosphate. So you usually think of ATP as the energy currency of the cell because it's high energy bonds within this molecule that get broken that enable enzyme steps to be catalyzed. So It's these high energy anhydride, phosphoanhydride, phosphodiester bonds that get cleaved and poof, release a cloud of free energy. And that free energy then is used to drive some sort of endothermic reaction within the cell. Okay, so ATP, remember its role not only as the energy currency of the cell, but also as one of the important nucleotides, which are found in what, DNA or RNA? Well, hopefully by now you're understanding that it would be found in RNA, the ATP nucleotide. So some of the key differences between DNA versus RNA are, number one, the ribose type. In DNA, you're going to find deoxyribose, whereas in RNA, it's just going to be simply ribose. That 2' carbon on the sugar is going to have a hydroxyl group in ribose, in RNA. The pyrimidine base is also going to be a key difference. So for DNA, you're going to see cytosine and you're going to see thymidine, whereas in RNA, you're going to see cytosine and uridine in the nucleosides that encompass that molecule. So remember, the differences are the ribose sugar and the pyrimidine base type. Here is a nice table which compares some of the nomenclature that's used to describe some of these nucleosides and some of these nucleotides. So you'll notice on the left here that the different base structure, adenine and guanine, which are your purines, and then cytosine, uracil, and thymine, which are your pyrimidines. Okay, so when you start attaching a sugar to the base, adenine becomes adenosine. When you attach a sugar to the base, guanine becomes guanosine. So for purine bases, you see that you get this osine, O-S-I-N-E ending. So anytime you see osine at the end of one of these, they're not talking about the nitrogen base, they're talking about the nucleoside itself. Now for the pyrimidines, you see that the cytosine, uracil, and thymine ending is replaced with an idine, I-D-I-N-E. So cytosine becomes cytidine, uracil becomes uridine, thymine, well, it would become thymidine, but you don't actually ever find that in the cell. Instead, remember, T's are found in DNA, so you'd only ever find it in the form of deoxythymidine. So notice the nomenclature differences as you go from a nitrogen base towards a nucleoside. Oftentimes in textbooks and in speech, people will use these interchangeably, but on the exam, they're probably going to be a little bit more specific, so you should know the difference between the base and the nucleoside. And then finally, as you start to attach 1, 2, or 3 phosphates to each of these nitrogen bases, we go from being a monophosphate, MP, to a diphosphate, DP, to a triphosphate, TP. And this pattern is used universally among all of the nitrogen bases, the A's, the G's, the C's, and the T's. Now, if you see a little D in front of one of the the abbreviations for one of the nucleotides, that simply means deoxy. And remember, which carbon is it that becomes deoxy, that makes something DNA versus RNA? It's the 2' carbon. Remember that 2' carbon. That's key in differentiating RNA from DNA.